Good evening, all. Today, my presentation is about integrating the Institute of Medicine's 6 quality aim into laboratory practice. As a lab professional, it is very important for us to integrate align our quality into the hospital requirements. So, as you know that the well-known standards of like medicine 6 quality aims by IOM, Institute of Medicine. So today I'm going to share our experience, what we are practicing, and what is the best practice to be followed in terms of laboratory, how to integrate the 6 quality requirements into the lab practice. My agenda for today is Institute of Medical Medicine's 6 dimension of healthcare quality, importance of classifying the dimension of quality, organizing quality measures by using quality domains in laboratory medicines, and at the end, discussion and quality question and answers. So, IOM 6 dimension of healthcare, as you all know, it is one of the well-established and well-practiced throughout the globe. 6 aims or domains of healthcare quality. So, it is applicable in we can apply this in all the sections or all the departments as a whole in the healthcare industry. And so the first one is out of 6 domains. There are 6 domains. The first one is safe, effective, patient-centered, timely, efficient, and equitable. So what is safe? A small introduction is given here. Safe dimension is about avoiding harm to the patient. So whatever we do related to intended to avoid patient harm is categorized as a safe healthcare quality domain. So effective, providing service based on scientific knowledge to all who could benefit and refrain from providing service to those not likely to benefit. So providing an effective care, doing the right thing and avoiding the wrong thing in general, doing the right thing as per the scientific knowledge. So that is an effective dimension of healthcare quality. Patient-centered, providing care, ensuring patient values guide all clinical decisions, involving patient in the care, providing confidentiality, privacy. So these all considering the benefit, keeping patient as a focus of care. So this patient-centered quality domains are designed keeping patient as a center of focus. Timely. So timely, the dimension of timely is to reduce waiting time or wait and sometimes harmful delays for both those who receive and those who give the care. So as you all know that timely care is very important in terms of healthcare. So this is also one of a very important factor of IOM 6 dimension, one of the 6 dimension of IOM. Efficient care, doing care in a proper way by avoiding the waste or reducing the waste. So whatever dimension is focusing on providing a care without waste is considered as an efficient care. Equitable, so providing care that does not vary in quality because of any extrinsic factor. So providing a uniform care regardless of gender, race, geographical distribution, or whatever, regardless of difference. So the care should be equitable and accessible. So in addition to this 6 dimension of healthcare, there are more dimensions being developed by different concepts. So let's say like access to care, appropriate care. So there are several, but these 6 there are some papers or some researchers have published based on the effectiveness of care given by the 6 dimension in the healthcare quality. So what is the importance of classifying the dimension of quality? Why it is very important for to classify ourselves as a healthcare professional? Because as we discussed, although there are more healthcare quality dimensions available, but the framework like the IOM domain make it easy for the healthcare workers to grasp the meaning and relevance of quality measure. It is to organize or it is to create, to practice our healthcare day-to-day practice in line with one of these IOM 6 dimensions of quality. So this will give us a clear direction that what we are intending for, how we are doing our practice. There are some studies which explore there are some publications and studies which explored that if the terms are understandable and resilient, researchers found that potential users of public reports responds well to a simplification of this framework into three categories. So there is an publications which talks more about this. If you see, using the IOM framework of all public reporting on quality, even Joint Commission International have published in their magazines, in their safety volumes, they have published that categorizing the quality into this 6 IOM framework is much effective and much relevant. So when we further classify, there is the categories can be further classified based on the care provided. So safe care, like care that provides patients from medical errors and does not cause harm. So that is the ultimate aim of a safe care. Whatever we do related to, for example, in our laboratory practice. So there are some activities we carry out to avoid safe, to enhance the safety of patients, or at the same time employees also. So those all we categorize as safety related quality domains. So care that is proven to work or get results, to get intended results, so which is effective and efficient. So whatever we for example in the laboratory, we try to give an accurate patient results. So that is effective and also efficient way of doing the patient care. Care that is responsive to patient needs and preferences. So always patient is a center of focus in any care or any healthcare settings. So patient happiness or patient satisfaction will complete this healthcare process. Regardless of what we do at the end, the patient should be happy or patient should be satisfied. So those all whatever we are intending to fulfill the needs of patients and their preferences, which this patient-centered, timely, and equitable quality domains will contribute to fulfill the patient needs and preferences. So those 6 dimensions of quality are further classified into these 3 to fit into these 3 categories. As I said, even Joint Commission International have a very elaborate presentation or very elaborate issue related to this using the IOM framework for all public reporting and quality. There is an recommendations. There is a recommendation from Joint Commission International to use the 6 quality frameworks to report all public reportings also. Also, there is one more article. Many articles are there, but for the presentation, I have taken 2 references. So even in pediatric critical care, it is concluded that if the care of pediatric intensive care patient is to be fundamentally improved, and understanding of current care environment, the existing evidence based, the opportunity for improvement and documentation for the improvement needs to be realized. The Institute of Medicine 6 aims provide a useful framework to advance the quality of care in the pediatric subspecialties. And perhaps other frameworks also will help, but in specific, this particular 6 dimensions of quality there are several publications, research have mentioned continuously about this effectiveness of applying the 6 frameworks, quality framework into the healthcare practices. So, out of 6 quality dimensions as per IOM, safe dimension is one of the highest high priority, or it's considered as a highly important domain, because considering the impact and outcomes due to healthcare incidents. So always the unsafe healthcare practices will lead to a lot of legal complications, reputation failure, financial loss, and also time consuming, and sometimes it is irretrievable also. I mean, we cannot go back and correct the errors. So since the safe care is a utmost priority in any healthcare organization, so whenever we are choosing any quality dimensions, so we may need to mainly focus on safe dimension in addition to the other dimensions of quality of care. So always when we choose a performance measures, so we need to give more weightage to the safe care, considering the other dimensions of quality. So what are the advantage of categorizing by quality domain? So what are the disadvantage? So the one of the main advantages, consumer responds positively to this categories. It is easy to understand, so easy to classify, and the outcome is also we can travel in a direction towards the 6 different quality aspects. And it is relatively easy to map these quality measures into these categories. So based on our day-to-day practice, we can easily classify. So any quality measures, any performance measure, we can put it into this one of this criteria. While choosing itself, we need to make sure that we are traveling towards, or we are trying to align one of these 6 quality measures. And any performance indicators or performance measure, we can add even more than one quality indicators. It can travel in more than one quality dimensions. This approach reinforces the IOM's widely accepted framework for quality. So that is also one of the main reason why one of the main advantage of it. Disadvantage, there are some disadvantage maintained in some according to AHRQ. The presentation is made from the AHRQ articles. So the disadvantages, categories are relatively unfamiliar to most of the healthcare professionals. So unless we as a quality professionals, we need to make sure that our staff understand why this indicator is chosen, what is the domain, or what is the rationale behind choosing this. And the next thing is, if you have very large number of measures overall, or in one of these 3 areas, you will probably need to find an additional way to break them into more manageable chunk of information. So when we have some areas where we need an improvement, so sometimes we may not be able to fit into one of this criteria. So in that way, we need to find still it is okay to choose a performance indicator, but depend upon that, but we need to mainly focus on choosing one of this indicator performance measure in order to get the more fruitfulness out of it, because it is a well proven methodology. There are several publications related to this who have researched on this topics, and also it is one of the main recommendations from IOM. So I would like to share our quality indicator monitoring data collection form from our what we are currently practicing. This is how it looks. So a department indicator title. So we choose what is the title of this indicator. Let's say an example, sample rejection rate. So what is the dimension, which dimension it is being aligned with? So we choose whether it is safety, effective, efficient, timeliness, patient-centered, or equitable healthcare. So specimen rejection, obviously it is related to safety, because specimen rejection may occur at different causes. There may be a different cause, like improper labeling may be in one of the main reason. Collecting in a different containers is also another reason. Sending it in a different transport containers, sending it in a different timings, insufficient sample. So when collecting a different sample, wrong labeling is also one of the reasons. So if we don't focus on this indicator, we may miss out that wrong labeling scenarios. So that may lead to a wrong entry of result to the patient, and obviously it will end up in a wrong treatment to the patient. So safety is one of the main concern. So we choose this as a safety indicator. Also efficient. So how good this process is being followed in terms of sample collection, labeling, and pre-analytical process. So all this, because this sample rejection happens in the pre-analytical phase of our testing. So this clearly indicates that how good we practice in capturing the errors and fulfilling the gaps find in that sample collection areas. So rational, every indicator we start with the rational, why we are collecting and how it has been going, how it is going to impact our or improve our work process. Selection criteria, so whether it is high risk, problem prone, high volume, high cost, or any other reason. So what made us to choose this, or why this selection is brought into what is what is the reason why we choose this indicator. Then data definition, what exactly we are monitoring, numerators and denominator, data outcome, how we are going to present this data, either in terms of percentage, numbers and all. So inclusions and exclusions also very important, because it has to be very clear to the users or the to all the stakeholders who is involved in this KPI monitoring, to understand the process, what all to be included, what all to be excluded. Then what type of measure it is going to impact, either it is a process, structure or outcome. So how does it is going to be aligned with the process of main quality elements. And then how the data collection, what is the main source of data collections, and what all the tools we are going to use for this, and what is the data collection methodology, whether it is like how whether it is retrospective or it is concurrent or it is prospective. So we need to choose what we are going to do. Then the target setting is very important, and what is the base of target, whether it's an internal target or is there any publications, is there a benchmark. So what is the reference behind it, who is responsible, and who is the data collection, who is going to do this task, then whom it is going to be reported, and then the monthly data, and there's a run chart where it displays the target and how it has been traveled over a period of time. So this is how in our setup, healthcare setup, we do the data collection before we do any new KPIs. So we need to fill up this form and make sure and submit to the quality department, and make sure that this form has been revised and approved before we start doing the data validation pilot study. We need to submit this to get approval. And this is an example of quality measure using aligning the quality measure using the quality domains in our laboratory. These are some examples of KPIs which we have, which we are practicing at present until last year. So sample rejection rate, blood wastage, blood transfusion reactions, amended and corrected reports. So here we define our targets, what is the frequency as well. If you see, we classify as per the dimensions of quality. It's not clear here because I took a screenshot and put it. So domains in our work practice, we have three. We classify or we divide our work phases into three phases: pre-analytical, analytical, and post-analytical phase. So pre-analytical is primarily from the patient taking the right test orders from the doctors, choosing the right time of collection, and identifying the patient in a right way, practicing all hand hygiene requirements, then making sure the test requirements or prerequisites are fulfilled, and then collecting the sample in a right way, right order, and right transport container, right temperature, properly labeled, and sent to the some samples. We have some criterias like after the drugs, before the drugs intakes, like therapeutic drugs, and some samples we have like fasting requirements, and some sample midstream collection of urine, sputum. So different specimens, so the criterias are fulfilled. So the domains in our laboratory standards, like we have some accreditation standards. As per that, we need to choose the domains. We need to fulfill some pre-analytical KPIs, analytical and post-analytical. And also it is very important to choose the healthcare IOM KPIs as per the IOM 6 dimensions of quality. So the dimensions: safety, effectiveness, timeliness, efficiency, and patient-centered. So these all we categorize it before the start of the KPI, and we put the monthly data. We highlight when there is a outliers from the target, or when we are getting away from the target, so it has been highlighted. So when there is a continuous failure, we take it as a PDCA to improve this project. So this is how we put it as a monthly dashboard. This is not maybe very appropriate way to display the data, but after understanding from the CPHQ class, so we are trying to improve it to give it in a best and more appropriate way of displaying the data. So these are all some examples of how we incorporate this 6 dimensions of quality into our work practice. So safe laboratory specimen rejection rate is included in the safe practice of safe dimension of quality. Then staff hand hygiene compliance is one of the KPI, because we have some main departments like highly infectious or prone to infection departments and work, and we are directly handling the biological samples, so the high risk of exposure. So due to that, hand hygiene is one of a very important requirement to monitor. Blood transfusion reaction rate. So again, considering the safety of patient, and so this transfusion reaction is very important in order to ensure that right blood groups and right antibodies related bloods are traced and screened before it is been given to the patients. Panic value notification. Again, panic value is a life-threatening situation, so we have a protocol to ensure that it is being given in the right way and communicated in a right timely manner to make sure that patient is unsafe manner. Effective physician satisfaction with clinical laboratory service is one of a way to evaluate ourselves whether we are providing effective patient care or not. Also, nursing satisfaction survey. Monitoring of frozen section, permanent section, correlation over a data over a period of time. So histopathology, like this is a one good indicator to make sure that reports are very effective in terms of comparing the frozen section and permanent section correlation results. Utilization of blood and blood products. So blood and blood products are very essential and very vital requirements. So we need to make sure that it is being effectively utilized. It's not over-utilized, or we don't run out of stock for emergency transfusion. So this is also one of our key indicators, one of our performance measure, where we evaluate our blood banking effectiveness. Amended or corrected lab reports. Due to analytical error or clerical error, sometimes we may end up in correcting the lab results. So there is a protocol for practice. There is a protocol for correcting the report and correcting it in a timely manner where avoid unnecessary treatment or wrong care to the patient. So this is also implement or reflect in our how effective the process is being carried out. Patient centered. We collect data from the patient satisfaction survey by focusing on different factors based on the patient, whether the patient care is being given with the rights and responsibilities, privacy, confidentiality, and whether it's a safe care, comfortable care, areas, how clean the area, how good the staff are behaving with the specimen collection, how informed the staff about what they are doing in the work practice. So these all we are collecting to evaluate and focusing on patient. And reducing patient waiting time is also one of not putting burden on the patients to wait for a long time to receive their care. So we are also monitoring it by some data collection methods to make sure that the patient waiting time is not extended. Timely care. Lab reports, it's like turnaround time is one of an example what we are monitoring in generally in the lab practice to make sure that the lab is giving reports on time to give to make the whole care effective. whole patient care effective, because 70% in general, 70% of clinical decisions are made by the physicians using the lab diagnostic reports. So giving a timely report is very important factor for a lab in order to maintain a good operations. Outsource report that. So sometimes we are unable to perform all the laboratory, all the tests requested by physicians by in-house due to limitations in methodologies, due to resource unavailability. So in that case, we outsource these tests to the referral laboratories. And in order to make sure that the quality is maintained, so we make sure that turnaround time, in addition to other factors, this is one of the key factors where we maintain a turnaround time, and we constantly reminding them or making them to report our results on time. So this is also focusing on the timely dimension of quality. Efficiency, laboratory document control practice. So there are some there are some mandates from the regulatory requirements globally, and as well from the accreditation requirements for a minimum retention and proper retention of laboratory documents. And sometimes it serves as a legal documents for the patients, especially in terms of DNA testings, histopathology, and blood banking requirements. So there are standard and procedure for the retention and archiving of documents. That how efficient these documents are maintained in each section is being classified as a efficiency of document control system. Even every single document's retention and destruction has been documented. So where we pull out the data to make sure that all these documents are retained in as per the defined protocol. So this exactly reflects on the efficiency of document control system. Blood culture contamination rate, mainly on patient safety, timeliness, and efficiency of the work process is reflecting in this. So blood culture contamination is again, it is a very crucial process in terms of treating the septemic patients. So in a timely manner, it has to be reported. So if there is a contamination during the collection phase, so it is very late to identify the contamination. So we have a very strict protocol for collection and monitoring this process. And this also efficiency, in addition to efficiency, it also reflects on safety of the patient. Equity measures. So in laboratory, as of I could not figure out anything related to equity. Maybe Sir can link something into it. But in a hospital setup, number of beds and types of services available based on the patient needs or based on the area or the geographical requirements. So that is one of the measures which I took a reference from AHRQ. And nurses to patient staffing ratio is also an equity measure. Percentage of physicians who are board certified is also one of the example of measure mentioned in the AHRQ for the equity measure of quality domains. So in terms of laboratory, we take some benchmarks and reference from different publications. Mainly College of American Pathologists, they have many references, and they publish a benchmark, they update on annual basis. So most of the data is what I have presented here are based from the QProbe lab benchmarks, as well AHRQ quality measures in the hospital settings are presented in this. And thank you all. Any questions?
Yes, I have one question, Mr. Kamurudeen.
Yeah. Yes, please.
Yes, one in the safe dimension of quality, you said the panic value notification. Can you explain it more, please?
Yeah. So panic value notification, because it's a panic value is basically designed on the life-threatening values only. So for example, like cholesterol is not life-threatening, so there is no panic value. Troponin, acute onset of myocardial infarction. So troponin is a first indicator to elevate. So this is life-threatening. If we don't report it on time, it may lead to a fatal injury to the patient. So if we delay the reporting, once the results are published, once the results are reported, so it is very essential for us to communicate to the doctor within 15 minutes of releasing the results. because that will have a doctor will make a decision based on this, based on timely care. So it will affect the safety of the patient. The care will be broken. So timely care is very important. As well, it will impact the patient safety. So that's why the panic values are not assigned to all the laboratory parameters, all measured parameters. We have more than 1,000 laboratory assays. So hardly we have for like 30 or 40 assays, we have these panic values. Prothrombin time, where troponin and potassium. So some values where it can glucose, like hyperglycemia. So patient may go to coma at any stage, like when it is more than 700 milligrams, or hypoglycemia less than 40 milligrams. So patient may go to a severe, untreatable or difficult situation. So that is how it is being linked with the safety aspect of quality domains.
So what is the total number of performance indicator in your department, sir?
Sir, we have 13 indicators. Sir. We have several sections actually for blood bank, hematology, biochemistry, histology, cytology, microbiology, special immunology, molecular. In the laboratory setup itself as one laboratory, so we have different sections. So we need to cover all the sections, and as well all the aspects of quality, as well all these dimensions of quality. Mainly we focus on the safety, sir. Safety to make sure that more.
How many indicators under safety, sir? Do you have any classification? Do you have any classification like number of indicator under safety, number of indicators under efficient, effective?
Right now, 5 is being linked with the safety, sir.
Safety.
But, yes, sir.
Okay.
Yes.
So whether you are calculating any score or compliance for each dimension, like what is the compliance regarding safety and what is the compliance regarding efficiency, because we need to measure the level of quality.
Yes, sir. But we didn't classify it, or we did not categorize it as that dimension, sir. Only identifying it, but at the end, outcomes are not classified into that aspect, sir. Maybe we need to incorporate this from this.
Yes, because if you want to know which one is, we need to focus first. So without this classification, without calculating the compliance, so how can we identify the opportunities for improvement?
Correct, sir. Yeah, we need to, we will be doing this.
Yes. And why still you're using bar charts, sir, in the dashboard?
Sir, that was the last year, like until December, but obviously we got.
Please change, please.
Yes, sir. I'm working on it to make it much better than this was the primary. For like we started last year, Jan actually.
Okay.
So
No
obviously we will be doing it as per the class now. Whatever I learned from CPHQ from your sessions, obviously I have a note more than CPHQ class. I will certifications. I will try to implement this practice in our day-to-day practice.
Excellent. Thank you, sir. And regarding your definition form, is it possible to show your definition form? I want to see your definition form.
Okay, sir.
Yeah, this is the one. Yeah, this is the one. So here, what about the sample size? Whether you are going to collect data from a random sample, or sample size is not applicable for this, because this is about sample rejection rate. So how you're collecting data? Whether you're collecting data from a random sample or from each and every.
Most of the indicators, all the samples are like, for example, like we have PT performance rate, we have sample rejection. So all the samples are collected, sir. But in terms of patient satisfaction rate, we are doing a random sampling, sir. We have a random sampling. In terms of nursing satisfaction, employee satisfaction, physician satisfaction, our focus is to cover all the employees, sir. For example, nursing, we have 700 nursing staffs. Doctors, we have 200 doctors. But we tried, not all 700 are reporting, but almost 400. Last year, we took 400 surveys.
Excellent.
But we didn't do any data sampling study, but this year, planning to do.
But better to include separate row for sampling, sir. Even if it is not applicable, we can put any. If it is not applicable, we can put any. But we should include separate row for sample size and sampling method.
Sure, sir.
And another thing, sir, this is sample rejection rate, correct?
Yes, sir.
Rate means we should multiply by 1000, correct?
Yes.
But as per your data, now you are calculating percentage.
Yeah.
And what is 103 percentage? Percentage 103 or 1.03? I think
1.03, sir. 1 point. So
Okay.
total
But
31,726 samples were collected. So in that 327 rejections, so 1.03%.
So here, what is the denominator? 31,000, 25,000, 28. So almost all are more than 10,000, correct?
Yes, sir.
So if you have more than 10,000, the denominator, we cannot recommend percentage. We should calculate rate. Rate means we should multiply by 1000.
Okay, sir.
And even the indicator is rate. So for rate, how can we calculate percentage, sir?
Yeah, yes, sir. Yeah. So this should be a sample rejection.
Rate. There is no mistake in your indicator, sir. But the problem, mistake in your calculation. We cannot multiply by 100, correct?
Yes, sir. 1,000.
If it is rate, if it is rate, what is the numerator? See, so what is your numerator here? Numerator, number of rejected sample, denominator, total number of sample received. And where is the formula?
It's here, sir. Yeah, the numerator is denominator.
No, we should create one more column for formula, sir. Formula is equal to numerator divided by denominator into 1000. Multiply by 1000. This is the formula, correct?
Yes, sir.
Numerator separate, denominator separate. Add one more row, formula, which is equal to number of rejected sample over total number of sample received into 1000. This is the exact formula.
Okay, sir.
So better to include formula column for each and every indicator.
Formula and calculation method.
Formula. After denominator, add one more row and include the formula.
Okay, sir. Sure.
And what about the benchmark? Benchmark is less than 2 or more than 2. It should be more specific, no? We should write either less than 2 because this is about rejection rate. I think it should be less than 2%, not exactly, We cannot write like this. Either we should write less than 2% or more than 2%.
Okay. Yeah, I think that makes.
So this is less than 2%, correct, sir?
Yeah, yes, sir. Less than 2.
And how can target as well as benchmark same? Target means we should develop our own target based on our data, not from a research article. We cannot refer research article for target.
So what we thought is we will do it for this year because every year we change the indicator based on the requirements. So we thought like we will collect our data, then we will while revising the KPIs every semi-annually we are revising the KPIs. So that period that time we will establish our own target, then we will proceed.
But already you collected data over a period of 12 months. So this is the right time to develop internal threshold.
Yes, this year we were modifying all.
Change your target also. And benchmark from, what is the source for benchmark, sir? Research article or from?
Research articles, NCBI. So they are publishing lab related.
So sorry to disturb.
Yes, sir.
So again, the benchmark and target should be in rate, no, sir?
Yeah, rate should be 2 per 1000, not 2%.
2 per 1000 means 0.2%.
0.02, correct?
No, sir, but here the NCBI gave 2% as a threshold. I mean, it's not target, like, so if we are converting into rate, then it should be 0.
Yeah, yes. But better to use only rates for each and everything, not only for performance indicator, even for target benchmark, convert into rate. Thank you, sir. And what do you think about effectiveness? For effectiveness, we need to use evidence-based practice, correct, sir?
Yes, sir.
So how you are establishing evidence-based practice in laboratory?
Evidence-based practice. Now we are following the let's say our lab is a CAP accredited and JC accredited. Also, whatever standards they are releasing, for example, related to hematology, we have International Society of Hematology Practice. So whatever SOPs, whatever practices we do in the lab, it is in compliance with that one. And also CLSI, Clinical Laboratory Standards for Standard Institute, is a global standard establishing committee for the laboratory practice. So every standard published is based on that one. So we are practicing whatever we try.
Mostly now you are focusing on accreditation standards, correct?
CLSI standards, sir. Accreditation is based on the CLSI standards.
Okay. Is there any collaboration with other organization to improve your laboratory? Whether you are doing any collaboration? Is there any agreement, collaboration with other organization?
We don't.
No, this is also we can do. No, sometimes we can collaborate with other similar organization to improve our performance. Correct. This is what we need to do for benchmark. We cannot compare with the NCP article. Correct.
Yeah.
Right. This will be, excuse me, this will be ENQUAS, right? EQUAS, the external quality assurance.
Yeah, in terms of ensuring the accuracy of testings and quality, because our outcome or our product is
right.
So this
that will be external quality assurance. We need to do that.
Yes. We have a CAP accreditation, CAP PT programs for every single analyte. So that serves as a quality assurance. That results being matched with the peer group, like 500 participants throughout the globe using the same machine, same methodology. So our results will be matched with them and we will be rated according to it, sir. So this benchmarking we are doing.
Okay.
Monthly.
Excellent. And whether you're publishing any papers from your practice, any research article, whether you publish any research article from your practice?
No, sir. We have a lot of abundant of data actually, but we tried, but last year only I joined, so we couldn't. Now we had a discussion to pull out the data, make it useful.
But this is very important for effectiveness. Without research, how can we establish evidence-based practice? Correct. First, we should learn from our data, from our practice, not from NCB or from accreditation agencies. Correct.
Yes, sir. We had an internal committee or meeting to start this project, like to pull out the data, put it together. We have doctors to do that and all, but we didn't take it forward, busy with so many other projects and all.
Is there any peer review committee for lab under your department, sir? Any peer review committee?
Yes, sir. We have,
especially for laboratory.
Yes, sir. We have a doctor for each section, head of the sections. Like we have microbiologist, hematologist, pathologist, microbiologist specialist. So when any disputes or any discussions related to that, not as a committee, hospital as a committee, but in terms of laboratory, we internally discuss. We don't have coined a term as committee peer review for the laboratory. But when there is a discussion, so doctors will take a decision. They will give their input and consultations.
Okay, good.
Sir, I just have a doubt on
regarding effectiveness of the laboratory test. I just want to add on saying that the effectiveness we are taking also from the standard guidelines like national HIV guidelines like NACO or certain guidelines to further to pitch in whether we are using the appropriate test. If the appropriate test is not done.
Yes, Dr. Savita ma'am. Actually, yes, we can focus on accreditation standards, even some clinical pathway, clinical guidelines. In addition to this, we should learn from our data. This is more important for evidence-based practice, for effectiveness.
Okay.
We should convert our data into practice as well as research. This is also we can to establish evidence-based practice. Correct?
Yes, sir.
Yes, Dr. Reshma ma'am. Yes, sir.
Sir, also like when Dr. Savita said, so I remember a point. Like like some data has been shared with the national, like for example, antibiotic sensitivity. So like there is an annual review by using the peer, like Dr. Savita is participating in it.
Okay.
I mean, at the hospital, so we share the data and we try to understand where we are standing on it, comparing our data. And same happens with MOH, with our molecular parameters, with our blood bank, with central blood bank. So, as an individual, there are some activities in section wise also happening. So.
Okay. Whether you are providing any education to your students, because you are in the university hospital, correct?
Yes, sir. We have regularly assigned the students, sir, like every semester. So on each section, so there is an rota for them, and we provide a training, not hands-on training. They cannot handle the patient samples.
Is there any education for quality?
No, sir. That is not in the module. The university is
this is very important, sir.
Yeah,
this is very important. As an academic medical center, we need to teach about quality to our students. Otherwise, how can they will?
When I inquire them about their syllabus, because we are just a practical, we comes into the part of a practical, the fourth year, like some semesters we are there. We are not a part of the syllabus as a part of a training. This one, sir. So we try because after our training, they go for the license, MOH license and all, because they get graduated. So we try to give but it is an informal way of educating them. So it starts with quality control, how we do and all. We have our own way of, we have our own checklist for educating the students with these all. But as a syllabus, they have some quality requirements in their syllabus. I have gone through one time about their syllabus, but academically, I think there is a syllabus, but we don't specifically go for the quality topics. We may need to improve that. Yeah.
Regarding TAT, is there any difference between male and female patients, sir? Whether you check the difference between male and female patient regarding TAT, turnaround time. This is one of the equity measure. Sometimes we can check, is there any difference between male and female? Is there any difference among the nationality?
No, sir. There is no. As such, nowhere it is reflecting. Once the
Because
sample
here we in our organization we found there is difference between Saudis and expatriates.
Okay.
Regarding TAT, there is significant difference.
Okay.
So,
so regarding this, generally, I think it's practiced everywhere. If the VIPs
that we need to check, is there any difference?
Those get processed earlier than the other patient sample.
So if the TAT time is less for VIP, what does it indicate? There is no equity in your organization.
Yes, I was just going to mention that would be an equity related indicator if there's a difference based on social or socioeconomic background of an individual or a patient.
Yes, we need to check, sir. Is there any difference in our demographic variable, not only for gender, even for other demographic variables?
But it's difficult to pull out who is VIP and all, sir. Maybe at one scenario now, for example, someone or doctor will call and as Dr. rightly said, so he's from like a royal family, so we need the result immediately. So, but our system is in such a way that stat samples are like, we have a dashboard running on every department. So the stat samples are given priority, and routine samples we have minimum of 4 hours of processing. So yes, as she rightly said, it happened in several scenarios, but that population may be very less. Yeah, that affects the equity, rightly.
Yes, even we can compare by different department, by among physicians also. Is there any difference?
Yes, sir. That we need to see.
Yes, sir. Yes, sir. Thank you, sir. Any other question from others, please?
Sir, I just have a query on what being told on peer review, sir. Sir, to my understanding and other things, peer review is something that a test or something, a diagnosis which is done by a one person is reviewed by another person. So actually for radiology, how we do is, if a Dr. A has reported the test, it should be reviewed by another doctor. It cannot be reviewed by him. So irrespective of one person have done the job, somebody have to review and say whether it is correlating with what person A has reported. So practically, if I say for lab equals, that is actually possible. I have done ethics and I've given a sample and then, but
Anusha ma'am, peer review we cannot recommend for each and every test. If there is some
yes, sir, I
abnormal,
agree.
if
Sir,
there is
but
some outlier, and if you want to validate that result, we can recommend peer review. We cannot recommend for each and every procedure and every test. Correct? Am I correct?
that's what I just wanted to understand, sir, because I don't have any ideas on cap and other things. So that's why I just wanted to get it clarified.
Am I correct, Kamurudeen sir? Peer review we cannot recommend for each and every scenario. Correct? For
It
each and every
cannot,
test.
no, it shouldn't be
But
for
I
each and
just
every,
wanted to know,
but
do they
randomly.
Yes.
Randomly, you need to do that.
No,
Yeah,
no.
peer
I just
review
wanted
is
to know
mostly
if the
important in
practice
case
there.
of histopathology. Yeah, it's mostly
Histopathology, I agree,
important in
ma'am.
case of
Usually, histopathology slides actually go
histopathology
for review and
reports. Peer
opinion.
review is recommended.
Yes,
Okay,
ma'am.
okay.
Histopathology, 100% agree. Even in India, we do it. But I'm
Yes.
not sure on the other clinical hematology and biochemistry or molecular from your end. I'm not sure whether it has been peer review is practically possible or is it recommended from?
Actually,
Yep. It
for
is
other
possible
department, we
again
have
for
competency. We
hematology
have
even.
competency
Yeah.
test,
Yeah, in all
not
the
peer
section,
review.
what all tests are there in our scope, we are doing the peer review. External QCs are there for each and everything.
External QC, I agreed, ma'am. I just asked only for the internal 16. External QC, I'm agreed. I'm not asking
Yeah,
anything
there is
on
a peer review in the external QC, no?
ma'am.
No,
Since
peer
the
review is
discussion
for
is going on,
like
I just wanted to
competency
know the
assessment.
practice which has been done in the other place. I'm not saying that this is required or not required. I'm not aware
Okay.
because Kamurudeen have added that they have gone with cap and other things. So out of things only, I'm just clarifying. Is
Yes,
it
Anusha
recommended?
ma'am. Yeah. The practice vary from organization to organization, but there is some
That is why
standard
I
for peer
just
review.
wanted to.
Yeah.
Yes, sir. I just wanted to get it clarified for my understanding. That's it.
Okay, good. What about Dr. Reshma ma'am?
Yeah, so good evening all. Um, it was a very good presentation, Dr. Kamrit. So I'm really, I'm not a lab person, but I always wanted to learn and learn more. And there are more topics which I would say request you to take on lab quality, lab QA also, maybe Westguard, maybe how if you have been have an opportunity to work on lean or six sigma, I think a lot of scope is there in laboratory. So yeah, it was very good to listen to how the dimensions of quality are can be depicted in a laboratory also. So good wishes to you. I'm very happy I attended this webinar because I'm not from lab and I want to know more about lab. And sir, one more thing was that about this peer review, even I felt was um, what EQUAS and BQUAS we do in our country. So I was under the impression that that is only peer review. But I understand now that histopathology is where peer review can be maybe easily or maybe practically possible. I don't know about other sections of laboratory.
Thank you, Dr. Reshma, for your nice words. Thank you so much. It's very encouraging. Regarding,
more sessions,
yeah,
yeah,
if,
बोलिए ना.
yeah. Sir, yeah, we can conduct more, and if SAR permits to do, we have some presentation. I'll be more than happy to do that. Okay. Regarding regarding the peer review, it is only when there is a human skill is involved, because if you literally see the laboratory analytical process, we have made most of them are automated process. And ensure the quality by having a stringent quality practice, as you rightly said, like we have Westguard rules and quality control for every step, right from the collection until there is releasing. But there is a human skills where it will have a great impact on the decision making. Let's say like smear study, blood smear interpretation, bone marrow aspiration study. So it differs from individual to individual. Also microbiology, some interpretations by the microbiologists, as well histopathology. So this individual skills when there is a microscopic skills are involved. So in the CAP competency, we have a peer review study, like they will send a slide, stained slide. It is considered as a patient sample, so we don't know the result. So it directly reflects when we report it back. So they correlate with the group of physicians, and then final interpretation will come out. So we can evaluate ourselves, the doctors, how good the research reports is been given to the patient. So it directly reflects. When I was working in India, there were several microbiologists or several histopathologists in the same place. So they go for a peer review. One interpretation will be reinterpreted by the other stuff, but it is limited to the availability of the same specialist and the same hospital. Or otherwise, we need to send out, means it has to be charged to the patient. So for second opinion, we have to charge to the patient. So practically, where I'm working now, it may not be possible. But still, sometime what happens is the physician themselves visit the lab to discuss, to see whether it is right, or whether the doctor reported. So they themselves, when they have a very good insight on this interpretation, so they try to come and discuss with the doctors, see the slide, and try to co-interpret together to understand like whether the results are done correctly or not. So that is also sometimes documented in our setup. Yeah, thank you.
So that's all we have got for you today. So if you have any more clarifications, you can mail us. And we are sure that we'll be doing more webinar from laboratories also. So related with KPI tools and everything. So thank you for joining today, and thank you all.
Thank you. Good night.